So next we're moving to Europe, and Joachim Lundeberg will talk about Swedish initiatives.
Thank you. I'll share a small vignette of the work that we're doing on developmental cell atlas. And this is a project that actually started through the funding of CCI, the Chan Zuckerberg initiative. And by that, we actually enable us to also to get some additional funding from private foundation in Sweden. So we have actually a 5 years project into this part of the Human Developmental Cell Atlas. And the people that we have, it's a star team. I'm a little bit biased since I'm part of it, but the other ones are stars. We have Emma Lundberg being a star in spatial proteomics. We have myself working with spatial transcriptomics, Mats Nelson working in cytosequencing, Sten Lindarsson working on single cells, having a lot of brain competence, clinical partners being Erik Sundström, and working with lung with Christos. And all of this is kind of handled by a scientific coordinator. And this is a fantastic opportunity for us because we're all sitting in the same site at SciLife Lab in Stockholm. So we are enabling to look at these precious material using this set of technologies that I will describe shortly. Obviously, this is one of many developmental cell atlas initiatives, and we are super happy to engage with them and find the ways to go forward. Still, this is early days, but I will share some of the data that we have. So we are focusing on 3 organs, being brain, heart, and lung. We're looking into the first trimester, which means the 4th post-conceptual week up to the 12th conceptual week. And we are actually using then a set of technologies to look at these samples, one being single-cell RNA sequencing, providing us with the single-cell clusters and the cell type annotation of that. And then we have this fantastic opportunity on the one and the same set of samples, query that with a set of different technologies, being then spatial transcriptomics, in situ sequencing, and spatial proteomics. And our end goal with these samples is actually to create 3D structures of the organs that we're investigating. So practically, we have this then opportunity to actually, from the same organ, make multiple omics from one and the same sample. So we actually, in practice, is that we do sectioning of this, we do spatial transcriptomics, and the next slide we use for in situ sequencing, then spatial proteomics, and so forth. So we iterate this, so we actually get a 3D view by multi-omics way on a single organ. All of the protocols that we use are actually are accessible at Protocols.io if you're interested. So the way that we thought about launching this project was really to use 2 data-driven approaches to select the clusters that we really would like to investigate further. So in one case, we have the non-spatial 10x kind of single-cell data, which is crisp and great to have the cell types being annotated. But the question comes quite fast, and where are these cells positioned? And then we go to our spatial methods, and then we're using spatial transcriptomics, and in this case we're using a barcoded glass surface that pulls out the RNA to get that spatial information of that. But actually also the spatial technologies are providing new insights to histology and to morphology of the tissues that we're investigating. So a number of these clusters of spatial gene expressions are actually trigger us to ask the single cell data set what kind of cell this is. So in all, we are doing data-driven analysis to be able to select the genes that we would like to confirm and work additional with the next set of technologies. So then we are then generating these data-driven clusters on the left side, and then we go into the other set of omics technologies, which is then in situ sequencing using Mats Nelson's technology, and then we have the spatial proteomics headed by Emma Lundberg, which is really using the Codex platform. But on top of that, we also have access to 18,000 human antibodies from which we could use. And these are then being obtained by the Human Protein Atlas. So I think we have a number of good tools to elaborate on these developmental organs. So I have to share some very recent data and just give us a flavor here how we are doing this. So this is some single cell data from the lung. On the right side, you see the ages that we have today, which is 3 ages, 6, 8, and 8.5 weeks. We do clustering of all these at the same time. But for lung, we have two compartments, right? So one compartment goes for single cell, and the other side of the lung goes for spatial analysis. So we have a fantastic opportunity here to just compare single cell data directly on the same individual and do a spatial analysis. And on the up here, you have the lung morphology at this 8.5 week stage, and we have then different distances along this organ. And then we, on the same H&E images, we are then performing spatial transcriptomics and using data-driven approaches. And we then see clusters of gene expressions that we could then compare with the single-cell data set. So we get a lot of things from one single sample. So this is just to remind me that it's a very important finding, well, not a finding, observation, which is very easy to think about it, is the temporal aspect. So we try to actually look into these organs over development, and on the down right here we just added the 6-week sample, and we get some clusters of gene expression that we could compare how this organ develops. And I think this is a very useful resource. So what is the status today? Well, we're focusing on brain, heart, and lung. And on the left here, you see the clustering of the entire data set from the 3 organs of the different ages. We have about 1 million single-cell data points. And the one that we have done most on in terms of using all the omics technologies is actually the heart. And these are represented here among the big clusters. And just taking that data, we could recluster them, and then we get a higher resolution of the single-cell data. And then we also then using spatial transcriptomics to identify spatial patterns, and combining this and this, we could then go into these validations, here being inside the sequencing for a number of genes. And what is very nice, and we have the possibility on the same heart confirm that by spatial methods, by spatial proteomics. And today we've done about 100 tissue microarrays, and this still is early days, but has kind of demonstrated that we go from a single organ to a multi-omics analysis. So with that, I'm done.
Thank you. 1 or 2 questions? Questions? Yeah, somebody in the back.
Hi there, thanks very much for your talk. I was just wondering how the sort of Swedish effort on the developmental cell atlas is linking up with other international efforts?
Yeah, that's a very good question. We have actually a meeting later this afternoon for the different efforts. So we have already started talking about sharing data as well as tissues, and I think these are still early days. So I think we will have much more of a community in this aspect.
Great, thank you very much.